critical for nearly all biological processes, but overall we lack a solid understanding of their significance inside living cells. So why is this a problem? Well, to understand function, we need to purify proteins of interest and visualize their dynamics and interactions. But to purify and visualize, this is challenging because it's difficult to isolate and distinguish individual proteins from other biomolecules inside the cell. So in recent work, researchers have been using genetic fusion tags and attaching them to proteins of interest for isolation, and they've been using fluorescent proteins for labeling, capturing. But the challenge with tagged proteins is that they're present at low concentrations and are not highly abundant. So they're limited by their binding affinity, selectivity, and kinetics. And this limitation is primarily due to the equilibrium-based nature of the binding between the tag and the ligand. So their goal is to have these tags have a reaction that's rapid, selective, and irreversible. And so that's where the Haloalkane Dehalogenases come in, which are hydrolytic enzymes that allow rapid and most importantly, irreversible attachments to synthetic ligands. And so the benefits with them is that they're small, chemically simple, and they're membrane permeant, which is ideal for live cell uses. Here they chose to focus on DHA because it had a broad substrate distinction, so it could interact with haloalkanes attached to a variety of functionalities. And they initially synthesized a panel of chloroalkanes containing FAM and TMR fluorophores, and then they modified DHA to form a stable attachment with chloroalkanes by trapping the covalent intermediate that forms between the DHAA and chloroalkane. And in doing so, they replaced histidine at the 272 position with 4 different amino acids to help prevent catalysis. And so in doing so, they found out that phenolphthalein reacted the most efficiency with the FAM ligand. And so this H272F ligand attachment was shown to have good distinction and stability. But the problem is that it had inefficient binding due to poor kinetics. And so next they focused on improving the binding efficiency of this H272F by taking a TMR ligand and docking it into the model, and a covalent bond was created to the asparagine at the 106 nucleophile. And then from a subset of amino acids that were 5 angstroms from the bound ligand, these 3 amino acids appeared to have the most contact with the ligand, and so these positions were substituted for their respective amino acid. And so when they implemented the substitutions, they finally got the halotag 2, which showed binding kinetics that improved 10,000-fold for FAM and 40,000-fold for TMR. But the problem was that it's essential to have a protein tag that's structurally compatible with the fused target protein. When halotag 2 was fused to the N and termini of a variety of proteins, the fusions expressed poorly in both cell-free systems and E. coli. And so the underlying cause of the poor expression was due to the inefficient folding because of the non-compatible structure. So to fix this problem, they realized they had to substitute the phenolphthalein that they previously implemented and replace it with an asparagine at the 272 position so that it could form a bond with glutamate at the 130 position. And then they did other substitutions like with D78G, so improve the thermal stability, and then 5 mutations to enhance expression, and then 1 for faster binding kinetics. And then they finally got halotag 3, which they examined by fusing it to the C termini of 3 proteins, and they found that it was beneficial to expression only in terms of C termini. For N-terminal fusions, the improvements were not as significant, And so they wanted to optimize this halo tag so it could be used as a general tag. And so they implemented 9 substitutions that were shown to improve the expression of soluble and functional proteins, and then they had 1 substitution to enhance the binding kinetics. And so they got halotag 6, which was then illustrated that it had an alpha helix at the C terminus that was in close proximity to the base of the binding tunnel. So this would affect stability and, again, the binding kinetics. And so they attempted to optimize the helix by random amino acid substitutions and introducing random two residue extensions. And so they finally got halotag 7. And so with halotag 7, the binding kinetics for both ligands, TMR and FAM, were further improved going from halotag 3 to halotag 7. And then when using 3 different enzymes, when halotag 7 was used as a C-terminal tag to an enzyme, the ID one, it provided 7-fold more functional full-length fusion protein compared to the original halotag 2. And then for 2 other enzymes, ID and Flunk, it was used as an N-terminal tag, and it was improved by 80 to 10-fold, respectively. And so overall, halotag 7, or just halotag, is a robust genetic fusion tag that uses an irreversible attachment to a ligand. It has a broad structural compatibility with fused partners, and it's small, so it's inert, and the binding ligand is chemically simple and compatible of carrying diverse functionalities, which enables protein capture and visualization. In this article, when looking at the X-ray crystal structures of 3 types of DHA, there's 2 tunnels that were found that connected the buried active site with the bulk solvent. So these tunnels being the main tunnel and the slot tunnel. And so the main tunnel is open in all 3 types of DHA, but the slot tunnel is only open in 1, and all the other types have the tunnel blocked. And so this brings up questions about the function of the tunnel and the role of the solvent for the release of the products. So what is the mechanism of the release of the products from the buried active site? Can we change the accessibility of the pathways by introducing mutations in the tunnels? So here they studied the releasing pathway behavior of 2 dehalogenation products of TCP, being the chloride anion, so Cl, and DCl. In their study, they had 8 mutants of the DHA carrying mutations at the residues lining the 2 tunnels, and the effects of the mutations on the accessibility of the pathways and the ligand exchange were analyzed and compared with the crystal structures of the wild type. And so 4 variants of DHA were from a previous paper, and they had mutations in the residues lining the tunnels, and then another 4 protein variants were constructed by site-directed mutagenesis and introduced additional variation in the 2 tunnels. All mutants were characterized for their activity with TCP, for binding of DCL, and for proper folding of CD spectroscopy. And so all mutants showed increase in the rate of TCP conversion except for mutant 14. And this was due to the fact that a bulky residue at position 176 was essential for enhanced activity, and that the highest increase in activity was determined for mutants carrying 2 aromatic substitutions in the main tunnel. As far as structural changes related to the mutations, mutants 4, 14, 15, and 31 showed similar intensity of their CD spectra to the wild type, so the structure wasn't affected, but the other 4 had changes in their spectra, so the structure was different. The structures of the enzyme product complexes for dehalogenation of TCP were modeled by molecular docking of DCl into wild type DHAA complexed with Cl, and the molecular docking trajectories were investigated for the release of the products being DCl and Cl, and were investigated for the exchange of the water molecules between the buried active site and the bulk solvent. And so the ligands can be exchanged between the buried active site and the bulk solvent by 5 different pathways and by 3 mechanisms. The mechanisms being the passage through a permanent tunnel, a passage through a transient tunnel, and then passage through a protein matrix. So the first pathway being P1, P1 being the main tunnel in DHAA, so the 2 helices of the cap domain with alpha 4, alpha 5, and 2 loops. P1 was open for only the wild type and mutant 14. And it widened during passage of DCl, Cl, and water molecules. And so the introduction of one or more aromatic substitutions in the main tunnel caused a change in the opening from a permanent tunnel to the transient tunnel. And so this just shows that accessibility of P1 was controlled by a number of things, but one being the aromatic residues, and then the varying length of the N-terminal of the alpha 4 helix, and then the flexible residue located in the middle of the alpha 5 helix. Pathway P2A was the slot tunnel, and this formed an open tunnel only during passage of DCl or water through P2A in proteins with I-135 or V135, so no mutation at those positions. And so P2A was accessible for DCl only in the wild type and mutant 4. For pathway P2B, this was the deep surface depression near the slot tunnel, and the accessibility of it was controlled by the aromatic residues, being the mutation of W or F 141 and P25F, as well as the flexibility of the NC loop. Pathway P2C was a branch of P2A and was formed by the NC loop, the alpha 7, alpha 8 loop, and was formed an open tunnel only in the wild type and the mutant 21, and only in the presence of Cl in the active site. And then finally, pathway P3 was not observed in any crystal structures of the wild type. It was located between the helix alpha 4, the NC loop, and the CC loop, and it was shortest and straightest compared to the other 3, but it was unfavorable for the water molecules and DCl due to the steric clashing. So overall, the main tunnel was open in all crystal structures of DHAA variants carrying the wild type C176, while a single mutation of C176Y blocked this tunnel. So 2 out of the 5 pathways, being P1 and P2A, were observed in the crystal structures, while all 3 other pathways were identified by molecular docking simulations. And the release of Cl was only in pathway 1, and the release of DCl was shown in all 5 pathways. And this release of DCl was enabled by the high mobility of the protein backbone and by progressive rotations of the protein side chains. And the point mutations introduced into P1 and P2A tunnels identified in the crystal structures modulated the accessibility of the individual pathways and led to a changed mechanism of the ligand passage in P1. So the accessibility and the mechanisms of ligand passage in enzymes with buried active sites can be modulated by mutations introduced into the exchange pathways.